Hi students, today we are starting our last topic of this chapter, that is chapter number 11, and our topic is transformation. For transformation with recombinant DNA is required because DNA being a hydrophilic molecule cannot pass through cell membrane. Hence the bacteria should be made competent to accept the DNA molecules. Competency is ability of a cell to take up foreign DNA. Methods to make a cell competent are as follows. Number 1, chemical method. In this method, the cell is treated with a specific concentration of a divalent cation such as calcium to increase pore size in cell wall. The cells are then incubated with recombinant DNA on ice, followed by placing them briefly at 42 degrees centigrade and then putting it back on ice. This is called heat shock treatment. This enables the bacteria to take up recombinant DNA. Physical method. In this method, a recombinant DNA is directly injected into the nucleus of an animal cell by microinjection method. In plants, cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA called as biolistic or gene gun method. Next, disarmed pathogen vector. When allowed to infect the cell, transfer the recombinant DNA into the host. Now we are starting application of recombinant DNA technology. It can be used for biological processes of cellular differentiation and aging. It can be also utilized in making genetic maps. It can be used to spell out complete nucleotide sequence of genome of various organisms including human. By this technique, useful compounds can be produced cheaply and efficiently. Vaccines for hepatitis B have been produced by recombinant DNA technology. Now we are starting application of genetic engineering. It shows the structure and mechanism of expression of genes. Operon concept of gene make the gene inactive or active. Introduction of a specific gene into bacteria or yeast cell is helpful in synthesis of human insulin, growth hormone, etc. Possible to introduce useful prokaryotic gene into eukaryotic gene. Introduction of gene coding for vitamins, antibiotics, hormones from higher animals to bacteria has helped to produce substances which are difficult to synthesize. It has helped man to use the therapy for certain hereditary disorders such as hemophilia. Large number of drugs are becoming inactive due to various microorganisms developing resistance due to uncontrolled recombinant DNA. Pathogenic microorganisms produced as a result of genetic engineering, if used in biological welfare, may be responsible for causing destruction of human race and other living beings. Now, gene therapy. In humans, it is used to replace a faulty gene by a normal, healthy or functional gene. So, today we have done application of genetic engineering and DNA technology, then different methods of transformation we have studied, and with this our chapter is completing. Today whatever I have taught you, I am sending you its PDF. Thank you.